Good evening, group. I'm Elsa. I'm a third-year PhD. I'd like to show you things that I'm thinking about and working on. And it's going to be about plant roots. So I work at Plant Sciences in the group of Julia Davies, and we look at how ion transport occurs across membranes, and we look at how ions are moved across membranes to then let the plant know what's happening on the outside and both on the as well as on the inside. So obviously, this is going to be a very colorful talk about all the things, or some of the things I think are pretty cool about plants in my area of research. So if you think about a career change after this talk, very happy to discuss it with you. Also, I wouldn't mind a pair of extra hands for my final year of PhD, so welcome to talk to me. And I'll start off with giving you the big picture, or rather the big problem that I'm thinking about. And then I'll zoom in into the smaller problem that I'm working on in the lab. Hey guys, welcome. And I also kind of zoom in because we're going to look at individual roots and a few cells at a time. Big problem. Or not problem, but what we know is that, or as the United Nations put it, that food begins in the soil. So pretty much almost everything we eat starts in the soil. So, or starts as a seed on the soil, the seed then germinates, grows roots. Those roots need to take up nutrients to make the plant healthy, to then produce a lot of yield, a lot of food for us to eat, right? If you eat a lot of meat, for example, you're still eating plants, if that makes sense, because that cattle is going to have eaten a lot of hay, a lot of soy, a lot of maize, which all grows on soil. And we know that having a very healthy and very fertile soil makes plants grow a lot better, right? So because we know that, we also know that putting on fertilizer, putting on more nutrients, makes plants grow even better, makes yields even higher, which we like. So when we talk about agriculture, or like our understanding of conventional agriculture, really we're only talking about the past 100 years, I guess. So what happened is we discovered that you could actually produce fertilizer, produce nutrients in a very high concentration, put them on the field, make plants grow better. In the 1920s, a mechanism was invented to pretty much extract nitrogen from the air, compress it down, and then apply it as fertilizer on fields. Yeah. The nice thing here is that if you extract something out of the air, air is pretty much nitrogen, or a lot of nitrogen in it, so we're not going to run out anytime soon. So we can chuck on the nitrogen onto the fields, we're not going to run out. However, the issue is another if we look at phosphate, which is the second most needed macronutrient for plant growth. So down here there's a bit of there's a graph showing how since 1900 through to 2010 we started extracting a lot of phosphate as phosphate rock, starting in the 1930s, 1940s, and then it's really skyrocketing. So more than 160 million tons of phosphate rock are extracted because that's the only way we can actually get phosphate to then process into fertilizer. So that has led to, some people call it peak phosphorus. There are different estimates around, but what it means that is we're taking phosphate out of rock, we're mining it to then process it into fertilizer to then apply to the field. And it's a one-way street, right? We had taken it out of the mountain, out of the rocks, to then apply to the field. Another issue that I wasn't aware of is, and I find very interesting in aspects of food security, is that rock phosphate occurs very patchy around the globe. So estimated 70% of it occur in Morocco, then you have a 5% in China, 3% in Algeria and Syria, like 2% in the US or something. So you might imagine that once we start to hit the ceiling with extracting too much phosphate rock and starting to run out of or deplete our natural resources, and this could become an issue because few countries sit on all the phosphate in the world. So I didn't put any numbers onto when people think we're going to run out of rock phosphate, Some estimates say 2050, which is fairly soon. Other estimates say, oh, well, you know, you didn't take into account whatever, it's going to be another 100 years on top of that. However, I think that's a very, very short time span altogether, considering that we so heavily rely on it. Like, you know, our agriculture builds on it. It builds on chucking on a lot of fertilizer. So what I thought is that, oh, it's in the news. We know that, you know, too much fertilizer is bad and this is what's happening. But I thought, I feel we don't really apply it to ourselves, right? Like it happens somewhere very far away that people chuck a lot of fertilizer on the fields, but what does it mean to us? So, who had a banana today? 3 people? I mean, how do you get to your 5 a day? All right. So let's take a banana, for example. I would like to walk through the whole fertilizer story looking at a banana. So what happens? Country A has a lot of rock phosphate. Phosphate is taken out, is processed to become fertilizer. This fertilizer is then shipped to country B, where if it's a rich country, they probably put a lot of phosphate fertilizer onto their fields. This phosphate either runs straight off, because a lot of the phosphate just leaches out of the soil into waters. It can also get stuck on soil particles, and really only a very little amount is taken up by plants, which is a huge problem in general, because plants find it very difficult to extract the phosphate out of the soil. So we have fertilizer on the field. Then, for example, we have our banana plant. The banana plant takes up phosphate through its roots. It transports it up. It uses it for everything, really. Puts it into the banana. Later on in the season, the banana is chopped off and transported, shipped to, let's say, UK, right? So you buy bananas, you take a banana, what you do, you peel it, you then eat the banana, and all the phosphate that's within the banana, you're going to take up, and then you have the banana peel, and you just put it in the bin. So what happens here? The phosphate that came from, you know, some mine in country A, and then was put onto a field in country B, is then shipped to country C, UK, and you then take it up, you throw away the phosphate that's in the peel, you take up the phosphate that was in the banana, you digest it, you might use a little bit of it, but the rest really just gets into sewage, water stream, whatever. In the end, I hope you can see that it's a one-way street. We're taking something out of a mountain and then putting it into water. We have runoff from the fields, we have sewage, and we're not good at all in, you know, taking phosphate back out of the whole system. So what happens is we're transferring it into waters. Could be fresh water, could be oceans, so leading to eutrophication, meaning that we have too many nutrients in a place where there should be very little nutrients. And it looks pretty, so what's shown here is a picture taken by NASA of the Baltic Sea, and what you might be able to see as this kind of green slush is cyanobacteria blooming. So things grow that shouldn't really grow in that abundance. And if you have a lot of things growing on the surface, they take up all the oxygen from the water. They also shade everything that's beneath. So that means the whole ecosystem is being changed to an extent where you just can't rescue it. And it's not easy filtering a whole ocean for the phosphate that's in it, right? That was the problem. Are you aware of it now? But now you might be, Elsa, but you know, why can't we just put something else on the field and make the plants happy? Why do we need to use phosphate? If we don't have a lot of it, why can't we just use something else? By the way, I forgot to mention that. Feel free to ask questions anytime, right? Comments, questions, go, fire. So, turns out we need phosphate because life itself loves phosphate. So it doesn't matter what we look at in a cell, if we look at DNA, RNA, our energy currency, ATP, if we look at our membranes that are pretty much just phospholipids, or if we look at our proteins that are, you know, switched on and off by phosphorylations, for example. Phosphate is everywhere. And there's really nice articles about it, you know, saying, oh, phosphate just has the perfect characteristics to kind of, you know, it's very stable, it interacts well, it can contain energy to a nice degree. So it's a very good element to kind of start building life onto. And when we look at what we think the first cell might have looked like, we're imagining something that had RNA on the inside and then being contained by a lipid membrane containing, being constituted of phospholipids. So already in the first cell that was formed at some point on our planet, we had a lot of phosphate in a cell. So phosphate and life go together hand in hand. Alongside with that is that life loves phosphate, but because of that, it doesn't really like calcium. Now the problem is that phosphate and calcium like to complex, and they complex really easily. So everything that we have in our body that's really hard, our bones, our teeth, is like pure calcium phosphate. So as soon as calcium and phosphate come together, they just, kind of stick together. It's very difficult to get them apart again. So I just told you that life relies on phosphate groups. And so very early in evolution, life had also had to take care of actually keeping the calcium in a cell very low. So, pumping the calcium out of the cell. So when we look at cells today, may it be plants, may it be humans, generally what we find is very high phosphate on the inside and very low calcium as well. On the outside, however, we have low phosphate and high calcium to prevent calcium and phosphate to precipitate. To regulate that, got a lot of PowerPoint animations. To regulate how much calcium we have on the inside and how much we have on the outside, very early as well in evolution, life had to not invent, but come up with a good system of restricting the flow of calcium from one side to the other side. So we have channels, we have transporters, we have pumps that are involved in maintaining a low calcium within the cell and a high calcium, or pumping the calcium out of the cell. This scenario also gives rise to something that occurs in humans, in yeast, in plants, and it's fairly well studied, so-called calcium signaling. Because we have so little calcium on the inside of a cell and so much on the outside, means that, transporting in a few calcium ions is a really good signal for the cell, ooh, something is happening on the outside. I might have to do something on the inside to change metabolism, to change gene expression, to, adapt to what's happening on the outside. So cells or organisms use calcium as a so-called second messenger. And because it's only one type of ion, you need to have a way of encoding information into just one type of ion. So what we find is that different stresses or different stimuli lead to different calcium, so-called calcium signatures. So you might have, a very high influx of calcium, then it's pumped out or transported out again, and you might see that repetitively. So this pattern or this calcium signature then leads to one response. Equally, you have a different stress leading to a different calcium signature, leading to a different downstream response. So that's just a really neat way of encoding something using one type of ion. Pretty neat, right? Okay, now comes something I've worked on all weekend. This is going to blow your mind. So, I told you that we have channels on cell membranes that regulate influx and efflux of, for example, calcium. Now in the literature we find, I looked into, ion channel. So how many ions do you actually shuttle across? So the numbers you find are 10 to the power of 6 to 10 to the power of 8 ions per second. Let's take, the average, that would be 10 million ions per second through a channel. Sounds like a big number, but you do physics and maths and something, and I guess, you look at stars and, big numbers aren't an issue. However, if, I try to actually think it through. So, 10 million ions per second through a channel. That channel isn't a huge leak in the, it's not a huge hole in the wall where everything just kind of leaks through, but it's actually very selective. So it means that only about 4 ions at any given moment can fit into the pore and fit through it. So, this is the image I came up with. Let's imagine a door, or that door over there, right? So I guess we could agree on how many people can you fit through that door in a second? Maybe, 2, 4, 6 people if they walk really fast. An ion channel works in the same way as a door, as it gates something. However, within a second, you would fit more than the whole population of London through a door. Okay, that's pretty cool, right? But that's not the whole story, because it's a door with a bouncer on it that, like, you know, whilst the ions are going through, it actually checks the ID of every ion, and if you're from Cambridge, you can't go through that door. All within 1 second. So, oh, right? How cool is nature? All right, so that shows you how within very, very short time, cells are able to respond to stresses very specifically by, for example, using different ion channels. As you know, I mean, if, you know, just in your brain right now, what's going on in there, and yeah. Let me now talk about what's going on in plant roots, because it's a similar thing.
Wait, I just have one
yes,
question.
please.
What kind of stresses would give these two different responses?
Okay, good question. I mean, those ones are just, they look pretty. They probably occur in a nucleus where we see oscillations occurring. What I'm looking at is, so the stresses I work with or that are relevant in the plant field would be, for example, salt stress, another osmotic stress, radicle stress, radical stress, such as ozone. You have pathogens.
Okay, but just so salt, if you put like some amount of salt in, then it changes, what is it, the rate at which ions go in and out of the cells, or?
So looking at calcium, applying salt to a cell would then lead to a very quick increase in cytosolic calcium and then a very quick decrease again, which is a different picture. I'm going to show you a few graphs later on, how you can, like in real time, see. I mean, you know, I know what stress I apply to the root, but even if I wouldn't know, I could tell you what stress the root responds to, because it looks unique. My research question, I'm going to show you 2 slides of data or so. So I talked to you about how phosphate and calcium have a very special relationship. I also talked to you about how phosphate fertilizer is a problem that agriculture looks into and should look into, and how it's very important for root growth in particular. Now, I do believe that we need to understand better how plants cope with too much or too little phosphate, to then, from there, find strategies to actually, you know, breed for plants that are better adapted to, for example, lower phosphate input. And so the detail I would like to add to this story is trying to understand how phosphate starvation, so a plant has too little phosphate, changes the way that plants perceive the environment by possibly changing their cytosolic free, so their calcium signaling machinery. Nobody's looked at that yet. And you already jumped one step ahead maybe, asking, you know, ooh, what does it look like? Because one of the tricky things about monitoring something in real time that's going on in a live organism is how do you make it visible? Visible in a way that, you know, our machines could actually detect it. And I just like to show you those 3 guys. They got the Nobel Prize in Chemistry in 2008. And Roger Jen was actually a PhD student at Churchill. Whoa! But not only that, those 3 developed so-called biosensors or reporters that we can transform into living cells and that enable us to actually see in real time what's going on under the microscope with, you know, other detectors. And what they did is they took proteins that we find in nature in jellyfish, Aquaria victoria. And I read the story of how Shimomura kind of, you know, told how in the 1960s, many summers in a row, his family and his colleagues would go down to the beach somewhere where there was a lot of jellyfish, and you know how they kind of, we would say they fluoresce or they, you know, shine a bit of a light, and they wanted to know what protein is behind that. So what they did is, like, summer after summer, used to kind of fish tons of jellyfish out of the sea, condense them down in some weird way, end up with a tiny, tiny amount of protein. And they then knew, oh, this is, you know, they had some way of knowing this is the protein that's responsible for giving us this luminescence. However, it didn't glow. It didn't do anything. And they're like, okay, so we need to add something to probably make it glow. So they tried different substances, and one of the substances was, seawater. And then they took the seawater and, adding seawater made the whole thing glow. So then they took the seawater apart, looked at the different components of seawater, and found that calcium is the one responsible to making this whole system glow. So what this jellyfish has is two proteins that are called aquarin and green fluorescent protein. So those three guys worked on identifying and characterizing and cloning those proteins. And what happens in the jellyfish is that the first protein, when it binds calcium, it emits blue light, so we call it bioluminescence. And so this blue light then is taken by a green fluorescent protein and turned into green light. And either of those two proteins are used to detect calcium in living cells. So I'm doing a lot of work with aquarin that's been put into a plant, and we also have a plant with different fluorescent proteins. We have green fluorescent proteins. So this protein has been engineered to bind different things and also fluoresce in different colors. It's all very pretty. Now the plant I'm working with, I call myself a plant scientist, but really if you ask me what the trees out there are, I wouldn't know, I guess. Because what I'm working with is pretty much one plant, so I'm not a botanist. However, I can tell you many stories about this one plant, which is called Arabidopsis thaliana. It's pretty much what mouse is, what mice are to human geneticists and human researchers. Arabidopsis thaliana is to plant scientists. So since the 1980s, it's very easy to work with. It's completely sequenced. It was the first plant whose genome was fully sequenced. It looks very small. It's a weed. It grows everywhere. Probably grows out there somewhere. You just would never notice it. But what we can do is we can grow it in the lab on agar plates, pretty much just like jam, where you solidify, something where you know the exact nutrient composition of, which makes it very easy to, know how much phosphate, for example, is in the solution, how much calcium there is. And we can also transform this plant with aquarin or other fluorescent proteins. And now coming to this, really kicked off an area of my project, and it's an area I'm still working on. I'm trying to, figure things out. So what I found is that this is going to answer your question as well, I hope, Jack. So in blue, we find a control plant which has been grown on, standard conditions. It has everything. It has phosphate and everything. And when we add a stress, we work a lot with ATP, extracellular ATP. Don't be confused. We have ATP in our cells, but it's also an extracellular signaling molecule. So, you might imagine a cell breaking open, the ATP gets out, then the neighboring cells should really know, ooh, something's going on here. We better do something. Something is wrong. It's danger, danger. So we work a lot with that and on ATP signaling. So you see that upon injection of ATP, I see an increase in cytosolic calcium, then a decrease, then another increase, a decrease again, increase again, and then it slowly goes back down to resting levels that are in the cell before the stress occurred. Now we know that this first peak is really a touch response. So when you touch plants, you will always elicit a calcium signal, a very steep increase in cytosolic free calcium. And this biphasic response back here is characteristic of a cell recognizing ATP. And now what happens if you take phosphate out of the medium, so the plants become very phosphate starved, you actually see they still respond to the touch, they show a first response, but then nothing happens downstream here. So that's been known for maybe, the biphasic response in response to ATP has been known for a couple of years. However, we still don't know the components underlying it. And because I found that under low phosphate, the signature changes, I'm trying to now take the different components apart and be able to nail down where in the root does the signal come from, what cell layer, responds first, how is the signal propagated. And using a fluorescent protein, thank you very much, Jack, for dimming the light. Using a fluorescent protein, we can actually look at those, look at increase in cytosolic calcium on the microscope. So what you see here is on the left, you have a plant root that's been grown on normal conditions, so including phosphate. On the right, a plant root that has been grown without phosphate. Light green indicates that, you know, it has resting levels of calcium, and the more it turns red, the more calcium influx there is. So I'll start the video, and you'll see after a while, I treated this root with ATP. You see a response, and then it slowly goes back down. So this is in a living organism monitoring cytosolic calcium. I'll play it again, and if I could now draw your attention to this area. So I'm sure you saw that there was a very sharp increase at the root tip. However, take a look at this area. So treatment applied and boom! Do you see that? Yes. Okay. And it kind of, you know, shows what I showed you before, that there's a biphasic increase upon ATP treatment. Now, if we look at the same, well, if we look at a plant that has been grown on low phosphate, treating with ATP, it responds again at the root tip. Very sharp increase in cytosolic calcium. Cytosolic calcium again is pumped out into the apoplast. And let's take a look again. And, you know, take a look up here if you see anything. Oop, sorry. Treatment. Nothing happening up there. So, yeah, I think you can turn it back up. Thank you. So from there, I can then extract data and, you know, look at which cells are missing a signal. I see that, so this is over time, and this is right at the root tip, further up the root, 2 millimeters up the root. What I did here is I kind of sliced the images, and each pixel is one image. So you can see that under full phosphate condition, upon treatment, you see a very, very sharp increase in signal in the root tip. And then later on, you see an increase in signal further up the root. However, if you have a phosphate-starved root, there still is a signal in the root tip, signal increase, however, no signal increase further up the root. So something is missing here in phosphate-starved roots. Somehow they do perceive the signal, however, the signal doesn't get propagated, or they've lost the ability to respond to whatever it is up here. So that's what I'm working on right now, trying to take the different components apart. You might be working with similar things. You know, I'm looking at what's going on, which cells do respond, in which order and when. If we take this as a schematic model of a root, you know, this is the plant. We take out a little section of the root down here. Does the occurrence of the different peak means that the signal spreads along the root? Does it mean that the signal spreads from the outside to the inside? Does it mean that I get a different signal from one cell, it's just, you know, different stages? Or does it even mean that I get a signal from the same cell again, but maybe intracellular stores are releasing calcium as well, giving me another signal?
But it was up the root that we were seeing that.
Yes. Actually, now that you say that, I realize that I'm already a step further. So this is from, you know, probably older presentation. So I do know that it's at different areas, but still you don't know which cell layer it comes from or which type of cell. This is another video that I took last week because we've got a new, maybe if you want to dim the light once more, sorry. And we've got a new reporter line in from Dale Sanders' lab in Norwich, based on the green fluorescent protein, which is a lot brighter than the other reporter lines I've been using. And again, this is just a standard root treated with ATP. I think now it's working. And you can see how it shoots up the root. And there's a bit of a secondary bump. And it's incredible how easy to use those fluorescent biosensors have become. Once you've got a stable line like that, it's beautiful to work with. So I'm going to work with that further. All right. I'm coming to the last slide of my talk. I hope I've showed you the bigger problem I'm working on and the details I'm trying to add to our understanding of how plants perceive phosphate starvation. And I'm really trying hard to fit this in because I just can't give a talk without mentioning Francis Darwin. Francis Darwin, son of Charles Darwin, who worked at the plant science department here in Cambridge. In 1880, he published a book, and he was pretty much one of the first scientists who realized that plants are pretty cool and do many things and aren't just like, rocks that are lying around and are dead and sad and, you water them and then they grow. So he did lovely experiments, well, not lovely, but very good experiments in our plant science department here in Cambridge, and published a book together with his father. If you're ever looking for a scientific historical read, definitely go for it. It's very nice to read. They just describe things so well. And also what they did throughout the book is they monitored the movement of leaves and also of roots of plants and show that, they spiral depending on night and day, for example, for leaves. So here is the trace of a root. They smoked plates, glass plates, and then had the roots growing down and then were following, the tracks they were leaving in the smoke on the plate over time. And I would like to finish by quoting Francis Darwin because he realized 150 years ago that there is no structure in plants more wonderful than the tip of the radicle. So that's been the root tip. That's what I'm working on, right? It's hardly an exaggeration to say that the tip of the radicle, thus endowed, acts like the brain of one of the lower animals. So plants are very, very much able to sense what's going on, and especially below ground, it would be the root tip that knows exactly what's happening around it. With this, I hope I could, tell you that plants are just very cool and are very much aware of what's going on around them. Next time you touch a plant, be aware that there's a lot of calcium being shuttled across everywhere, and many other things happen that we can't even see yet, probably. Think about the bananas you eat and all your vegetables and everything, really. I think my project isn't going to solve it, but I believe in talking about it and being aware of it, we can, change things and need to change things. And please talk to me about anything related to my topic, to your research topic, because there might be overlap, not sure. Or if you've missed all the ultimate frisbee practices so far, please come and talk to me as well. So with that, thank you. There will be cheese. Oh, fun fact. I didn't know that, but I came across it in my phosphate calcium research, that the reason why cheese gets softer is because it becomes more alkaline over time, which means that the calcium phosphate in the cheese precipitates out on the cheese surface. So enjoy cheese and wine. Thanks, guys.